Let's continue our discussion of DNA and the central dogma. So if you remember from the last video, we talked about how the central dogma outlines how biological information is transferred from a gene in the form of DNA to RNA to a protein that gets expressed either as a pigment that might give us hair color or eye color, or an enzyme that might help us digest lactose. So we're going to look at the first process, DNA replication. So last unit we talked about how the structure of DNA, the double helix with its complementary base pairing and anti-parallel structure, makes it relatively easy to transfer this information. And let's look at why that is. So when we replicate DNA, when a cell has to divide, make copies of itself, basically what happens is we unzip the double helix and new nucleotides will come in and bond with the nucleotides that were on the existing sugar phosphate backbones. And as we know from the last video, that if we've got an adenine and A, what will always bond with that? Well, a thymine or a T. So A always goes with T, G always goes with C. So it becomes relatively simple to fill in the new side of the ladder and get two new copies. So if we follow through these 4 steps quickly, we'll start up number 1 at the top. So here the parent DNA molecule has 2 complementary strands of base paired nucleotides. Step number 2, those parental strands will unwind and separate at several points along the DNA, forming what are called replication forks. So that's going to be right in here by the number 2. Okay, now at each fork, the parental strand, and we've got 2 of them, one side and 2 side, provides a template that attracts and binds complementary bases, always A with T and C with G. And finally, the sugar phosphate backbones of the daughter strands, the lighter blue ones here, are closed. Each new DNA molecule consists of 1 parental and 1 daughter strand. The result is which is called semi-conservative replication. And if we look at it very simplified, and we're going to unwind that ladder, and we're going to look at a very small piece. So over on the left under A, here is our parental DNA, and it's a ladder with a complementary base pairing. So the first step is to separate those 2. So we'll unzip it just like a zipper. Now each parental strand serves as the template, and you can see the lighter blue, the complementary nucleotides right here, will come in, and because of the hydrogen bonding, A with T and C with G, they fill in where they're supposed to go. And at the end of that, we get 2 new strands, each identical to the original parental strand in terms of the sequence of base pairs. But because half is the dark blue parental and half is the light blue, the new daughter strand, we get what's called semi-conservative replication. So the new strand isn't entirely new nucleotides, it's not entirely old nucleotides, it's a mixture of the 2, and that's why it's called semi-conservative replication. Now, a little bit more complex and a little bit closer to what's actually happening in the process. So if we look over on the right hand side, here's our double-stranded DNA. So the first thing that happens is we're going to be looking at a lot of different enzymes, and if you notice, all of these are going to end in ACE. Okay, so these are all enzymes that are going to help with DNA replication. So the first one, topoisomerase, basically is going to kind of stabilize the double helix and allow it for the first big one that we want to talk about, helicase, to come in. And helicase comes in and it splits the strands apart. So it unwinds them so that single strand binding proteins can come in, and they're going to stabilize those single strands because they're kind of floppy, and they'll stabilize it so that the next enzyme, primase, can come in. And what primase does is puts a little kind of starting signal. It puts a little sign on the DNA that says start here. And what comes in next is this other enzyme, DNA polymerase. It's going to make a polymer. And so what it does is it comes in and it looks for that starting sign put down by the primase, and it will add nucleotides 1 at a time in the direction, as we learned in the last video, 5' to 3'. So one of these fragments will always go in a continuous line towards the replication fork, and we call that the leading strand. Now the lagging strand has to be copied in the opposite direction, in this case moving away from the replication fork. And so a new primer will come in, maybe right here, and it will copy it out until it bumps into the next one. Well, it can't copy any further until more unzips. And so the lagging strand gets copied in these small bits and pieces called Okazaki fragments. And at the end, the last enzyme that we'll look at, DNA ligase, comes in. And what DNA ligase does is it comes in and it ties together, right there, it'll tie together those breaks in the Okazaki fragments, so we get one long continuous. So the leading strand is always copied continuously. The lagging strand is copied discontinually in Okazaki fragments. Now I know I just threw a lot of stuff at you right there. I'm going to show you a video at the end that will make it hopefully a little bit easier to follow. So the enzymes that we need to look at again real quick. So we've got the single strand binding proteins, SSBP. They stabilize the unwound DNA. Next, we've got helicase that comes in and it unzips the DNA so that we've got two strands to serve as the templates. Primase then comes in and it puts down a starting sequence. It tells us where we want to start. The DNA polymerase then comes in and adds nucleotides. It does the majority of the work in terms of elongating the complementary sequences. And then finally, ligase comes in and it ties the loose ends together. It kind of links the sugar phosphate backbone together on the lagging strand. And a little bit more simplified, here's a replication fork. And as we can see, as we go in right here, the leading strand, which is on top this time, is copied continuously in the 5' to 3' direction towards the replication fork, and it goes in one continuous stretch. The lagging strand down at the bottom will be copied in these small Okazaki fragments that ligase will then come in and tie together. So this shows a replication fork. Now, let's look a little bit more closely at primase. So what will come in, primase will come in and it'll put down a short section of nucleotides that tells the polymerase, the brown enzyme down here, where to start. And so the polymerase, the primer is now the starting signal. The polymerase then goes and can add nucleotides, again 5' to 3' in a straight line. And then the last thing that we didn't mention before is a different DNA polymerase will come in and it will replace the RNA primer with DNA so that it's all DNA. So we now have a complete daughter strand. And here's just a summary. Again, you can take a look at this. I'm not going to spend the time to go over everything, but this provides a good picture of what happens and the steps. So if you want to pause it and kind of read through this by yourself, you can, because it does. Again, it shows all of the enzymes involved. So we have helicase, we've got the single strand binding proteins, we've got primase, DNA polymerase, and finally ligase. So take a second and look at this as we go through. I'm going to take this time now where I'm going to show you a short video or animation of the whole process that'll make it hopefully a little bit easier to understand. And here we go.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions, and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
So hopefully that clears things up a little bit. It's not an easy topic, and it's one that you're going to have to kind of probably look at a couple times and spend some time on. And please don't be afraid to ask questions during class as well.